guys, so welcome to our science lesson with Educators 2A. Are you excited for a fun learning session? So let's delve in. Our topic for today is about the 6 steps of recombinant DNA technology. But before that, what is recombinant DNA technology? Recombinant DNA technology involves using enzymes and various laboratory techniques to manipulate and isolate DNA segments of interest. This method can be used to combine DNA from different species or to create genes with new functions. So now let's go to the 6 steps.
The first step in the recombinant DNA technology is the isolation of genetic material. In this step, the desired DNA sequence or the desired gene needs to be isolated from its source. So, example of the source is the human cell. This step is important because it ensures that DNA is free from other components like protein that could interfere with the subsequent steps.
Restriction enzyme digestion. Restriction enzymes are like molecular scissors that recognize specific DNA sequences and cut the DNA at these precise locations. This creates fragments of DNA with specific sticky ends, short, single-stranded overhangs that can later be joined to other DNA fragments with complementary sticky ends.
So let's proceed to the third step of recombinant DNA, the amplification using PCR. Making copies, PCR or polymerase chain reaction is a technique that allows for the rapid amplification of a specific DNA sequence. Enzymatic replication, it involves repeated cycles of DNA denaturation, primer annealing, and DNA synthesis using DNA polymerase. This results in a large number of identical copies of the target DNA.
The fourth process is delegation of DNA molecules. Joining fragments. The isolated DNA fragment and a suitable vector like plasmid are cut with the same restriction enzyme. The cut vector and the DNA fragment are then joined together by the enzyme DNA ligase, forming a recombinant DNA molecules.
The fifth step is insertion of recombinant DNA into a host. The recombinant DNA is introduced into a host cell, usually a bacterium. This process is called transformation. Making cells competent, host cells are often treated to increase their ability to make a foreign DNA.
Isolation of recombinant cells. Selecting the desired cells, not all hosts will successfully incorporate the recombinant DNA. To identify cells that have taken up the recombinant DNA, the vector often contains a marker genes, such as antibiotic resistance, that allows for the selection of transformed cells.
So again, the 6 steps of recombinant DNA technology are: first, isolation of genetic material; second, restriction enzyme digestion; third, amplification using PCR; fourth, ligation of DNA molecules; fifth, insertion of recombinant DNA into host; and lastly, isolation of recombinant cells. That's all for today's video. I hope you learned something. If you did, like and subscribe.